Hello, welcome. My name is Bruce Bryan, and I am here to talk to you today about what we are calling resources for hands-on labs in a hands-off world. We here at miniPCR Bio, like everybody, had to sort of reformulate how we look at science education starting several months ago. And I think everybody's learned a lot in that process. And today we want to share with you what we've learned and the resources that we've come up with to better support you, teachers, in this new reality that we all find ourselves in. If you're not familiar with miniPCR Bio, we make biotechnology equipment. Specifically, it's our goal to make accessible scientific equipment that really anyone can use, from students to professional researchers. And as part of our goal to make science accessible, we invest a lot of time in innovative curriculum that can really open up areas of biotechnology and molecular biology to student learning in ways that were previously inaccessible. I'm part of the curriculum team at miniPCR Bio, and I also have a research background, and I taught high school for 9 years. I see it as the goal of our curriculum team to develop labs and activities that really engage students in authentic scientific exploration, and that's what we want to try to share with you today. So, as I mentioned, 2020 has really upended everybody's plans, and certainly ours as well. So today I want to talk to you about how we as a company have responded to the new demands of teaching and teaching laboratory science. And I'm going to take you through a number of activities and resources that we have made available to teachers for different models of remote learning that are going on today. And as we go through this, I'm going to start with more higher investment models where teachers and students are finding ways to perform whole molecular biology labs outside of the classroom. And then we'll work our way to very simple things that you could just download off our website and use in class tomorrow. So first I'm going to talk about portable equipment. This here is our P51 fluorescent viewer, but we also make our miniPCR thermocycler and our blue gel electrophoresis system that we'll talk about. I'll also introduce you to programs that have worked with this equipment to bring it directly into the hands of students in their homes. I'm also going to introduce some activities that we're calling At Home by miniPCR Bio. These are intended to be engaging activities that can be distributed to students at home so they can have simple yet high quality hands-on activities if the laboratory is not available to them. Next, we'll talk about our Genes in Space program. Genes in Space is a completely unique science competition where students design experiments to be done on the International Space Station. We're able to do this because miniPCR Bio equipment is a central piece of the molecular biology workflow on the ISS. The competition is completely free, and along with it, we have a number of resources for teachers to implement it in their classroom. It fits so well with today's virtual learning because while it asks students to think deeply about science, it actually involves no hands-on lab work. And finally, we'll talk about some of our free resources. These include our DNAdots series, other worksheets that you can go and download from our website today, and also our webinars and free videos that we have on our YouTube channel that both explain science to students and teachers as well as take students through the laboratory activities that we provide. So that's the plan. Let's get going. At miniPCR Bio, our company is dedicated to producing tools for science and scientists. And by that, we mean anybody engaging in science, whether they're a middle schooler holding a pipette for the first time or advanced researcher. We do this by making affordable, accessible, robust lab equipment. Most specifically, what we have here, what we call our DNA discovery system. This includes our miniPCR thermal cycler and our blue gel electrophoresis system. These two pieces of equipment really revolutionized laboratory equipment that's available for classrooms when they were released, in that they are high quality equipment that's simple to use, affordable, durable, and also instead of having singular large pieces of equipment in the classroom, you could have several of these in the room that you can distribute among lab groups. PCR and gel electrophoresis are two tools that serve as the central workhorse to most any molecular biology lab. And our equipment really gave students hands-on access to these tools in ways that wasn't previously possible. I'll also be talking about our P51 fluorescence viewer. This is a small handheld blue light illumination system that allows you to view fluorescent molecules in a unique way. Now, as I said, these are tools for science, and while we certainly find a home in classrooms across the country and even the world, we also have been the choice for many years for folks that need to take tools to remote locations. So much so now that we sell, for example, our lab in a box here. This is a little suitcase that you can take your equipment with you on the road. And the truth is, when you make small, affordable, robust equipment like this, people take it to places that you would never expect. So, for example, Dr. Elaine Guevara brought her equipment to Madagascar to study lemurs in a field station where there's no electricity. Because our equipment is small and low power, she was able to run it off batteries and charge those batteries using solar panels. Dr. Ian Goodfellow, during the Ebola epidemic in 2015, brought an entire lab of very expensive equipment to Sierra Leone. But in that West African field environment, a lot of that really expensive equipment failed. miniPCR thermocyclers were able to get his operation back up and running. And even today, with COVID-19 testing and testing for other viruses such as Zika, people have taken the miniPCR thermocycler and our P51 visualizer and developed tests that can be done rapidly right in the field. And then to what is probably the most remote laboratory there is, the International Space Station. In 2016, the miniPCR thermocycler was sent up to the space station, and since then has been central to the space station molecular biology workflow. And we've been very proud to spearhead a number of experiments that have been done there in space. Finally, in the classroom, however, we know the classroom today doesn't quite look like this. It's changed a bunch. So one of the first things that happened when schools were closing in the spring of 2020 was that people who were familiar with our equipment, as their classes became remote, contacted us and asked, how can we send your equipment to students in their homes? Obviously, this approach takes a fair bit of investment just in terms of effort, distributing electrophoresis and PCR equipment to students at home, sometimes across the country. And so we started working with these teachers directly, some from college, some from high schools, on an individual basis. And in these early efforts, we had a lot of success with students doing whole labs, labs that would typically be done in the classroom at home, using equipment that's being mailed to them or delivered by their teacher, using it and then sending it back. One of the reasons that this was successful for us is that we have a fair amount of experience sending our equipment to naive users and working with them remotely to get it working. We've done this as part of our loaner program through Genes in Space, which we'll be talking about later. We give 2-week loans to classrooms where we mail this larger lab in a box around the country. As part of it, teachers get a full set of biotechnology equipment, the curriculum and labs they need to work with their students, and also the training and support with videos that they need to be successful. This is a free program. We do it just for the cost of shipping, which we've waived in need-based cases. So on a slightly different scale, this is something that we were kind of already doing. I'd like to just take a quick aside, just to put a little plug for the program. It's something that you can sign up for if you want to be eligible for it next year. For the Lab in a Box program, we mail 4 complete sets of equipment, including the miniPCR thermocycler, blue gel electrophoresis system, and all the accessories, equipment, consumables, everything that you need to do labs with at least 2 classes. If you're interested, you can scan this QR code to visit the website and request a kit. This year the loans are accounted for, but you can apply for next year starting in April. But again, that was just a bit of an aside to say that we have experienced shipping equipment and labs, often to folks who have never used such equipment before, and providing the support to help them be successful. So exactly how this model works is dependent on the teacher who's running it. We've had some folks who distribute the equipment, and people are more on their own to work through it using the videos and the guides that we have. And others who have done this as sort of lab group work, such as this snapshot from a Zoom call. where these are college students from around the country all doing the same lab together in real time. I'd just like to note that this teacher also sent their student lab coats, which I think is a particularly nice touch. Now, this school year, we have continued to work with teachers, again, to implement different types of learners or more intensive work with students at home. And I just want to share one example of this. We have partnered with Mass Insight and the Massachusetts Life Science Center, two local nonprofit organizations. We're located in the Boston area. And with them, we're shipping 250 AP biology students across 8 Boston high schools, basically this set of equipment and 3 different labs that can be done with it. This is a program that's underway currently. I'm going to show you some of the activities that you could do with this setup a little later coming up. But the big takeaway is that Boston currently is not in school. And so these AP biology students are taking a class that's supposed to have a significant portion of the time spent in class doing labs. And obviously, if you're learning entirely remotely, that's very difficult to do. So we're really excited that using the P51 and the miniPCR, we've been able to help provide students really high quality, full labs that they can do it completely at home. So these tools for remote science are now in use with teachers as tools for remote education. And while this is, again, a little bit more of a higher investment from teachers in distributing materials and organizing it all, we've seen it be really successful. Now, with the at-home by miniPCR bio line of labs that we've just released, we take that idea of sending equipment and reagents to students at home, and we've made it as simple as possible. In this line of products, we have 3 activities ranging from building basic lab skills like pipetting to genuine open inquiry using fluorescence as an indicator, and finally engaging in one of our most cutting-edge labs where students get to work with DNA and RNA and protein directly as they visualize transcription and translation in real time in a cell-free system. So let's start with micropipetting at home. These activities all come in a bag, about just like this, with all the resources and the reagents you need. And one nice thing that I like is that we put all the instructions and everything you need in a little handy-dandy little activity booklet with all the instructions printed up. We did this so there's no concern about compatibility of file formats or worry about students having a printer at home or trying to do lab work off a computer. It's all printed up for students to look at directly. Micropipetting at home takes students through the basics of learning the important lab technique of using a micropipette. We use simple colored dyes to practice pipetting, starting on what we just call our pipette practice card. This is just a plastic card, and with it comes instructions for a little activity. But the basic idea is that, I'll just show with my pipette here, on a card like this you can pipette up and down very easily. You can practice putting things down, picking things up, moving them around. With the activity, we have you use several different dyes, mix different colors, practice measuring different volumes as well. We also distribute a little silicone practice gel electrophoresis gel. This is a practice gel, but if you want your students to return to you ready to go in biotechnology, one of the harder things that they have to do sometimes is load a gel. And with this, they can just practice anywhere. Again, this comes with a full activity guide leading students through loading gels and finishing with them giving themselves a self-assessment. We then have our, the sort of culmination for this is our pet by number micropipette art. So we give you petri dishes and some templates in that booklet where students can do some micropipette artwork with the colored dyes that we send. This comes with some templates to follow and also gives students the freedom to create their own artwork at the end. I'll show you in this little video, but this part of this really works almost like an adult coloring book where you sort of get into a little bit of a zone as you're doing it. And I can just promise you it's fun to do. You start to get into it, and if your students pipette like this and do a few of these, when they return to you, they're going to be experienced pipetters, able to do real lab work without error. We'll let this finish up, and hey, look, it's a Tweety bird. There's several pieces of artwork that students can do, and again, they can create their own. So next, I want to talk about our second activity, which is uses the P51 molecular fluorescence viewer to explore fluorescence in an open inquiry approach. Students are provided transfer pipettes, tubes, a plastic grinder, some fluorescein. Fluorescein is a fluorescent molecule that comes in solution, so that they can explore fluorescence as a phenomenon and also as a biological tool. So one question is, why talk about fluorescence at all? First of all, it's fun. When you put something in the P51 and it lights up, there's something just exciting, almost at like a primal level, about seeing something glow brightly. But it's also a really interesting phenomenon, and it's been developed into an incredibly important biological tool. So, for example, here on the left is a jellyfish. That's where the original green fluorescence protein, or GFP, was isolated from. Fluorescent proteins are common in animals such as jellyfish, corals, and sea anemones, where scientists hypothesize that they serve different functions, possibly such as attracting prey. In biotechnology, that power of fluorescence has been harnessed, especially to visualize microscopic structures. For example, this image here, a few different fluorescent molecules have been used to highlight different structures in cells that otherwise would be just impossible for us to see. So quickly, I want to talk about how fluorescence works. So when I talk about the specific activities, we have a common understanding. This is the molecule fluorescein, which is what comes with this kit in solution. Fluorescein is a pigment, and as a pigment, it absorbs light, which means that when light hits fluorescein, an electron in the molecule jumps up an energy level. In other words, the molecule becomes excited. Fluorescein best absorbs blue light. So when blue light shines on fluorescein, an electron becomes excited. But electrons don't stay excited. The electron that goes up an energy level will eventually come back down. If you've ever worn a black shirt on a sunny day, you understand what happens with most pigments when that electron falls back down an energy level. The sun hits the pigment, the energy is absorbed, and then when that energy is released, it's released as heat. But in a fluorescent molecule like fluorescein, when it absorbs the energy and that energy falls back down at an energy level, that energy will be released as light. In this case, green light. But always the light released is a longer wavelength than what was absorbed. So for this reason, most of the fluorescent molecules used in science absorb ultraviolet or blue light, and then they release it as a longer wavelength like green, yellow, orange, or red. So one of the first activities to do with the P51 is just to realize that a lot of things fluoresce and explore that. And we ask students to ask questions about what they see. For example, in the activity we call Home Glowin, students check out some honey, olive oil, or milk, all of which fluoresce to some degree. And I guarantee, because it happened to me, once you ask people to start putting random things from their kitchen to see if it fluoresces, they don't stop. I can personally attest that not only does milk fluoresce, but so does mayonnaise. I've tried all the oils in my house. I'm going to show you that in just a second. I've done my honey, my ketchup, my hot sauce, half the herbs in my cabinet, all with varying success. But that's part of the goal of the lab. Specifically, we ask students to make observations and then ask a question they can test, and then to test that question. So, for example, from some simple research, I learned that olive oil is supposed to fluoresce, and also that nicer olive oils are supposed to fluoresce more brightly. It's believed that's due to the chlorophyll in the oil, which is something that we're going to talk about next. So I asked the question, do different olive oils and different oils fluoresce differently? So here's my P51. It's on right now. Right in there, if you can see the tube in there, I have a negative control. That's just water. So I went to my olive oil, which is labeled extra virgin olive oil, though I'll let you know we don't buy very expensive olive oil in this house. So I'll try it out. And if you see that, you can see it's definitely fluorescing, not too brightly, but it's fluorescing a red color. So I had a more expensive olive oil. It was like a dipping olive oil for eating with bread. And so I said, well, I bet that one will fluoresce really brightly, so let me try it. Oh, that's the wrong tube, actually. This is the one I'm looking for. So is this one. And whoa, look at that. So the interesting thing here is almost all that fluorescence in there comes from this little dot in the bottom. That dot in the bottom is an herb that was mixed into the olive oil. What I thought was my expensive dipping olive oil, when I looked at the ingredients, it's actually like half canola oil. So that kind of was disappointing. So I tried canola oil. It doesn't fluoresce at all. And then finally I tried my grapeseed oil, because my grapeseed oil looked green, kind of like olive oil. And I said, well, maybe it will fluoresce the same way. And that's what I put in earlier. And look, it fluoresces a completely different color. So something that totally different is going on there. So that's just to say that by asking interesting questions, students can get interested in this phenomenon and work to solve interesting problems. So I mentioned chlorophyll. We're going to look at that next. As I'm sure you know, chlorophyll is sort of what I think of as the heart of the electron transport chain in the chloroplast. In the photosystems is this molecule, chlorophyll, which is a pigment. Chlorophyll absorbs light, and then when it absorbs that light, it transfers it down the electron transport chain. So this chart shows the wavelengths of light that are best absorbed by chlorophyll. So you can see that, chlorophyll B specifically absorbs best around wavelength 460 or 470, which happens to be the wavelength of P51. Chlorophyll A also absorbs in the blue light range, but a little better in the UV. And then both also absorb a little bit in the red, but we're not going to shine that color light on them. So our question for this lab is, what happens when chlorophyll absorbs light, like a blue light, but it's not in the electron transport chain? We've taken it out of the electron transport chain. And this is interesting to look at, but it also really helps illuminate the function of chlorophyll as a biological molecule. So I'm going to demonstrate this here. The lab asked you to take a little piece of spinach. I didn't have any spinach in my refrigerator, but I did have a little piece of cilantro. So I'm going to put the cilantro just in a little microcentrifuge tube like this. I'm then going to add some acetone. So this is 100% acetone, just sold as nail polish remover. It cost me $2.39. I bought it from a few different drugstores, and I've never paid more than $3. So I'm just going to use my transfer pipette here and add, a bit of acetone. So maybe a little more than that. So there we go. And then I'm going to use my plastic grinder that comes with the activity to just mash this up. You just put it in, twirl it around, and you'll see that this is going to start turning pretty green here pretty quickly. So there we go, some nice green color there. So next I'll take just an empty tube, and I'm just going to, again, use my transfer pipette to add some to this tube just till it's, about half full. Perfect. I'll cap this up, and we're done with this right now. So in my P51, I'm going to put a negative control. This is just some plain acetone that I've prepared in advance. I then have a tube where I put a little bit of cilantro leaf in there so that we can see what happens when a whole leaf is put in. Now, as you can see, not much. And so then I'm going to put in my chlorophyll. Look at that. Look at how brightly red that fluoresces. That's just fun to look at. So again, the chlorophyll now, it's not in the electron transport chain. So the light is hitting it, it's absorbing that light, and then it's releasing that light back out as red light. The chlorophyll in that leaf is doing a similar thing. It's absorbing the light, and then it has to release it, but it has an electron transport chain to release that light into. We'll just look at that. We'll just look at that for one more second. It's pretty fun. So again, the chlorophyll absorbs blue light and will release red light. So finally, as part of our at-home series, we present the BioBits at-home Central Dogma Lab. This is a smaller version of our traditional Central Dogma Lab, but it comes with the P51 fluorescence viewer and also the option of adding an inexpensive fixed volume micropipette. This allows you to do the entire lab just with this pipette, and it's very inexpensive and can be used at home easily. The Central Dogma Lab allows you to visualize transcription and translation in real time. We use a cell-free reaction. Basically, that means we've taken all the innards out of cells, we get rid of all the cell membranes, and then we freeze dry those. So then when you add water, all those ribosomes, enzymes, etc. wake back up. And that means that when you add DNA and water, all those ribosomes and enzymes get going, and they will transcribe that DNA into messenger RNA, and then take the messenger RNA to make protein. The trick is that we've engineered into the RNA that is produced a signal that will fluoresce green. And then when the mRNA codes for protein, it codes for a red fluorescence protein. This means that in real time, you can watch RNA be made as the tubes turn from clear to green, and protein get made as the tubes turn from green to red. I really love this lab because it is a truly cutting-edge technology that we've adapted to show students one of the most fundamental concepts of molecular biology. And before this lab existed, I really don't know of another lab that demonstrates the central dogma to students in a way that they can actually manipulate. So to show you what this looks like, this image here is the P51. It's just a little dark so you can't see the front of it. This is a time-lapse video taken over about 8 hours. Typically, you would set up the lab, make an observation 15 minutes later, and then either 8 hours or the next day, come back and see the final results. So you can see here the tubes are not fluorescent. In the middle, we have a negative control, and then on either side, there are two tubes in which we'll run the experiment. So now, remember, we've added DNA to these tubes, and we've engineered that DNA so when the mRNA is produced, the mRNA will fluoresce green. So let's watch. You can see that relatively quickly, the tubes fluoresce green as that messenger RNA is made. Now, that messenger RNA codes for a red fluorescence protein, So as the mRNA is transcribed, the tubes will turn red. So let's watch that again. Right? After about 15 minutes, we start to see our messenger RNA as green, and then several hours later, we see the protein get made and it turns red. For the actual lab, we have manipulations to this. So if you change the system in different ways, students can predict how changes to the system will affect the production of mRNA and protein. Just to wrap this up, this is a really easy lab to do. It just takes a little bit of pipetting, but your students can use cutting-edge biotechnology to work with DNA, RNA, and protein all at home. They can watch the production of RNA and protein in real time and visualize essential processes of molecular biology. So that's the highlights of our at-home labs. As we go forward, we may look to add some more activities to this, but we really think it does a great job of introducing basic lab skills, inquiry approaches, and high-tech cutting-edge science to students outside the classroom. So next I want to talk about our Genes in Space contest. As I mentioned, our equipment is on the International Space Station, and we invite students from grades 7 to 12 to design experiments that can be done to address the challenges in space exploration. And every year, one of those experiments is performed by an astronaut in space. I mentioned our thermal cyclers on the International Space Station. That's it there, floating in the cupola of the ISS with a pipette and some tubes. So we're really proud that not only is the miniPCR thermal cycler on the ISS, but also that the first PCR experiment ever done in space was designed by Anna Sofia Bogorayev, a 16-year-old girl from New York, shown here on the right. In this picture, you can see Anna at the launch of the rocket that took her experiment to the ISS. Since 2015, we've had 8 winners in the Genes in Space contest. These are either individuals or teams who designed novel research to be done on the International Space Station. One of the things that we love about this contest is that it really challenges students to think big. We're asking students to design a DNA experiment that addresses the challenge of long-duration space exploration. It's a great example of how scientists approach these gigantic questions, whether it be spaceflight or even COVID-19 today. It's these collaborations of many scientists, each answering their small part of a much larger question that drives science forward. And students really do develop novel cutting-edge science. For example, in our 2018 teams, shown down here on the lower right, the winners, David, Rebecca, Michelle, and Arti, were interested in DNA repair in space, as radiation damage to DNA is a concern for astronauts. So they developed the first ever CRISPR-Cas9 experiment to be done on the space station. This is the same technology that just won the Nobel Prize, and here we have high school students introducing CRISPR-Cas9 to the workflow of the ISS. Now, I'm describing this as like really big, thinking, cutting-edge ideas, but the process is really easy to take part in. And it's extremely well-suited for distance learning. The submission is an online form that's a series of 5 questions, and each one is capped at just about 200 words. There's no hands-on lab component. If students can think of a good idea and they can describe an interesting experiment, they can take part in this contest. Of course, we don't expect students to just come up with great ideas out of thin air, and so we provide a lot of resources to help your students and help you work with your students to put together a really solid project proposal. This includes videos highlighting current research done in space. This includes live webinars with our Genes in Space team to answer questions along the way. And it also includes lesson plans and worksheets that you as a teacher can introduce to your students, helping them understand the biotechnology that's available for their use in the contest, and also just to get their creative juices flowing and to seed some possible ideas of the type of questions that could be asked. So I've highlighted some of the winning proposals, but I just want to acknowledge that every year we get hundreds of these proposals, and each one is read by several members of our teams. And the level of creativity and the amount of thought and scientific thinking that is put in by students from middle school through high school is always just really impressive. We really believe this contest is a method to engage your students in authentic, open-ended scientific inquiry, and we really encourage your students to apply. The next contest will open in January 2021. So right now is a perfect time to start thinking about what questions you could ask and to start working with your kids. Just a little added bonus, there's 20 different ways that we honor top proposals, and for every one of those, the teacher involved in it wins some equipment. The final set of resources I'd like to introduce to you today are free resources that you could download and implement in your class tomorrow. First off, over the past summer especially, we spent a lot of time and effort developing videos and webinars that folks can use in their classes, and we're continually making these today. We have some really high quality videos teaching PCR and gel electrophoresis, as well as teaching you how to pipette. In all of these, we have worksheets and activities that are free to download that go along with these videos. These ones are short, less than 10 minutes. You can implement very easily for students to watch at home and to do the entire thing at home. We've also done a number of webinars similar to what you're watching now, except we focus on one lab. And those we walk you through an entire lab from beginning to end, including all their curriculum. The idea being that maybe you as a teacher are interested in implementing this later, or maybe you want your students to follow along with a lab that they can't do because they don't have access to labs this year. For all of these, we offer a Google Doc-based worksheet that are free to use, and students can answer questions along the way as they follow along. You can also use the lab guides on our website. All of our lab guides are free to download without any type of purchase. So you can just go to the product page, download the lab guide, and see if it's right for you. I mentioned PCR and electrophoresis worksheets. I really want to call these out because I believe these are really high quality activities that we produce that are just free to download. They're just on our website for you to use. If you're teaching biotechnology, I know how hard it can be to find quality resources that are both comprehensive and the right level for a high school student. And that's why we created this. The final resource that I want to share is called our DNAdots. DNAdots are simple explanations of modern genetic technologies. The basic idea is that we take genetic technology that's maybe too new for a textbook, or maybe a hair above what would normally be included in a high school science class, and then we describe that technique in under 2 pages in high school friendly language. These again are free to download and cover a whole array of different topics, from directed evolution to CRISPR-Cas9 and qPCR and many, many more. And they all come with questions. We have always 5 review questions, 2 critical thinking questions, and then a discussion question at the end. We will also provide an answer key upon request, but we ask that you email us because we know you don't want those answer keys living out there on the internet. So just to wrap up, what I talked about today, we talked about portable lab equipment where students can do whole labs in their homes. We talked about the at-home labs, sort of smaller activities directly designed for students to implement outside of the classroom. Our Genes in Space program, a completely free contest where students can take part in cutting-edge science on the International Space Station. And then our videos, worksheets, that again are completely free to use, but provide an excellent resource for implementing biotechnology in a remote format. To finish up, I really want to thank you for spending your time with me today. I really hope that we at miniPCR can bring biotechnology to your students in a way that is useful and meaningful, whether you're in the classroom, doing some sort of hybrid model, or teaching remotely. Thank you for what you do this year especially, and thank you for joining me.